Protein synthesis is a two-step process involving the transcription and the translation phase. In transcription, RNA is made from a DNA template strand inside the nucleus. First step is initiation. An RNA polymerase attaches to the DNA at a specific sequence of nucleotides called the promoter. Polymerase unwinds the DNA at the beginning of the gene. Next is the elongation stage, where the RNA sequence is produced from 5' to 3' direction. New nucleotides are matched with their complements on the DNA strand, G with C and A with U. These new RNA nucleotides bind together to form a complementary copy of the DNA strand. Finally, in the termination stage of the process, the new strand encounters a termination sequence of bases called the stop codon. mRNA goes through multiple post-transcriptional modifications, such as the adding of adenine nucleotides, a 5' cap, and mRNA splicing. Introns are removed with the help of SNRPs, and exons are bound to each other, after which the mRNA leaves the nucleus through the nuclear membrane.
The process of translation is composed of three phases: initiation, elongation, and termination. The amino acid tRNA binds to the P site of the small subunit of the ribosome and begins to scan the mRNA for the start codon AUG. This process is called scanning. Once it recognizes the start codon AUG, the large subunit comes in and the process of elongation begins. The amino acid group of the tRNA breaks off and binds to the amino acid of the next tRNA with the help of peptidyl transferase. The ribosome then moves down the mRNA in the 5' to the 3' direction until it reaches the stop codon. Once the ribosome reaches the stop codon, there is no corresponding amino acid, and therefore the protein release factor comes in and stops the process. This is known as termination. This causes the polypeptide chain to break off and then assume one of the four structures: primary, secondary, tertiary, or quaternary, depending on its use in the body.